skipjack
push
tuna
that
is the
stick all
third
the way
one.
forward.
Yeah, so that was
Other
pretty
way.
cool to get to see them so active on that volcano site. There was a huge population of those skipjacks in
So
particular.
now you can back
Those
off
are the
on
trevallies that we
forward.
That's
saw.
a trevally?
Pull
Okay.
down in.
And those are the
And when you get
blackjack
about 20% on forward,
variety. There's
hit the button on
about a
top of your
dozen
right
or
joystick,
more
and that'll stick lock it.
trevally
Or you can
species
sit there and
in
play
our
with it
waters, so
to figure out how it
that
works.
was one of them, and they were following us around the entire dive.
Right.
Slow down
Yeah.
a bit so
Pretty
you catch
amazing.
up.
Yeah.
Anything you would say about that competition, Sean? Are you getting good feedback from the community? How are people
keep
responding?
pull down
There's
in,
a
100%
QR
down
code,
all
I
the
think,
time.
on the Fish and Wildlife website. I think voting closes on the
Keep
30th.
that left stick full. Oh, you're locked in now. Okay.
Or
Fine.
sooner.
Sorry.
I'm not sure, but
I didn't see
it
you hit
closes
stick
this
lock.
month,
Yeah.
and so the public gets a chance to pick one of those 3 fish as our state fish.
You
We
gotta
have
talk
a
louder.
state bird, we have a state
And you don't
plant,
have to
state
hold your
flower.
microphone to talk.
We have a lot of no state fish, so it's been
Yeah.
a long time coming.
That's,
So yeah,
interesting that that's one of the last to get added, right? I mean,
Yeah.
there's tons of birds too, but
yeah.
And it should be
Lots of
comfortable
fish, so-
talking.
Yep.
And if
Cool. And then I think
SPLs
that's
come in,
corresponding with
just
the
turn 'em off.
Fishers Week. So they're gonna, kind of
I
honor the
click off-
fishermen and
click
the
up on
fisher
SPL, then you
women in
hear
our
'em.
region. There's a proclamation signing and a whole week of
But
activities.
I keep talking
and
on 'cause there's
choosing a state fish
microphone
is part of
on,
that. So that was pretty cool.
yeah, it's a great process, and it's, I mean, your office,
Well,
the monument is, a huge part of that, building that for education and outreach for, the students in the region, and also, participating and helping EV Nautilus with this expedition as well. So we're grateful for all your work. And now I'm kind of thinking about state fish. I don't know if we have any trivia buffs, but anyone want to say what their state fish is? If you identify particularly with one? It could be a bird if you don't have a fish, or a plant, or really anything that identifies you with your state.
A state rock.
We're all quickly Googling our state animals.
What is the state bird for the CNMI?
It's the Mariana fruit dove.
The fruit dove, okay.
Yeah,
Not the booby.
not the booby.
Yeah, not the red-footed booty.
Yep.
All right, so Jacob, what's your state and what's their fish? I know you just have it off the top of your head. You definitely didn't Google it.
Oh, yeah, totally. For Rhode Island, the state fish is a striped bass. For Washington State, it's a steelhead, which I did not know. It's pretty cool.
I'm surprised Washington isn't the salmon. I wonder if Oregon is the salmon.
Yeah.
The
Something. I'll look that up right now. Oregon. Chinook salmon for Oregon.
If anybody on the internet, you can share with us. It doesn't have to be in the United States either. We know we have tons of viewers joining from all over the world, so you can give us a shout out for a fish that represents your place or another thing that you identify with. So the Pacific salmon of British Columbia, Verena, this is the provincial fish.
Yes, that's why Washington can't have it, because British Columbia got it first.
So much for the Salish Sea, huh?
Not only the Pacific salmon, British Columbia has claimed all 5 species of Oncorhynchus.
Now, Aaron, another video engineer, had a good way to remember the 5 species. I'm trying to remember it now. Ed, do you know? There's a good way, like a rhyme almost. It uses your fingers.
I remember thumb, chum.
Thumb, chum, yeah, that rhymes.
I think she just looks at her tattoos.
Cheat sheet.
Yeah, she's got a cheat sheet, permanent cheat sheet. No, I don't know. I'm not a fish person, so. Seattle's kind of known for salmon, but I think like down into Elliott Bay and some of Puget Sound, our biggest biomass is probably rockfish.
Yeah, or the gooey duck. I was thinking I always identified with the gooey duck in Washington State.
Shout out to
as well.
Evergreen State College, their mascot, the gooey duck. Not to be confused with Santa Cruz and their banana slugs.
Banana slugs.
Yep. Is that UC Santa Cruz or CSU Santa Cruz? I think it's UC.
I think it's UC Santa Cruz.
Yeah. Yeah, they're the banana slugs.
So we have some online viewers piping in. We have someone from Denmark who says the national bird is a mute swan.
Wow.
That's really cool.
And in New Zealand, this is going to challenge my pronunciation, kahuai. I think that's a fish, I want to say. National fish in Sweden is the perch. They have really good herring in Denmark and in Sweden, Scandinavia. The pickled herring is my go-to. Put that on a piece of small bread. It's like a very, kind of grain-heavy, lots of good stuff mixed into that bread. A little bit of, sauce on that, and oof, that's an afternoon right there.
Love eating forage fish. They're all very good, tasty, delicious.
Are we basing the greatness of a fish based off of its taste or what it looks like?
Yeah, that's an interesting question in the sort of national fish conversation, because is it just the one that you like to eat the best, or is it something greater? There's an overlap there, maybe.
I wonder if Oregon is the sturgeon. There's some giant sturgeon there.
I was looking it up, and Massachusetts state fish is the Atlantic cod. I feel like that sounds like one of those based off of a eating thing.
Right, a lot of cod on the menu, yeah.
I also looked up California, because that's where I'm originally from, and the state fish is a Garibaldi, which I've never heard before this moment. And apparently it's one of the fish that is protected, so endangered or at least not commercially fishable. So not an eating fish. Very interesting.
We're not too far from the bottom.
I looked up Alaska state fish, and it's the Chinook salmon. So
everybody wants the Chinook. It is a beautiful fish.
So Southeast Alaska and the Vancouver area can, have similar state fishes.
Right. Well, it's all Cascadia.
Yeah.
It's all one love for Cascadia. We have a viewer from Brisbane, Australia, and the barrier reef anemone fish, also famously known as Nemo.
And it might be Brisbane, but I don't remember.
Very iconic.
Yes. Thank you, Ed.
We have anemones and clownfish on the reefs here, but not that specific one. Yeah.
Right, the clownfish is a very, when you see it, anytime I see it in the reef when I'm snorkeling, it always reminds me of that movie. I don't know. It's such a distinct look.
Trade out.
Yeah, you never see the same orange with the 3 white stripes like they have in Australia or the Indo-Pacific. I saw one in Australia before, and it looks very distinct and very noticeable. It was really cool seeing them in the water.
Apparently in Victoria, it's the weedy sea dragon. Weedy sea dragon, yeah, that's a nice name.
They have a bunch of wolf eels up there too that are super cool to see.
Those are the ones that with the big forehead?
Yeah,
Yeah.
big,
Or
very
big
large
mouth?
head.
Yeah.
You can actually dive right in Victoria. Where is it? Ogden Point has a lot of them there in the rocks.
Yeah, I don't know what I'd do if I come face to face with one of those types of fish.
That's kind of like the eels that we imaged last night on the dive. They have this mouth opening behavior that looks aggressive, but is just to intake water.
They eat urchins, right?
Yeah, I believe so.
Yeah.
Those crushing jaws.
Yep.
So we are nearing the bottom here at the Burke Vent site.
Yeah, they can be.
So we'll let our
challenging
operations team in the front take over comms as they get us down to the seafloor, and then we can talk a little bit about our objectives and what our plan is for the dive.
Roger. We'll do all our bottom checks, and then we'll take the Neskins. We're starting to get beams, so let's take the stick out of there, and I'll go manual as well. Which way do you reckon is uphill? Or which way are we going to go, north? Yeah. I think we go, we start in the south and go north. Yeah, right.
So we're doing a bit of a zigzag search pattern on this little southern ridge.
I always ask this when we come down, I like to have the ROV on the shallow side to paddle into. Oops, wrong button. You're going to find the hydraulics are all wonky compared to the last dive, so we got them all slowed down for the shallow water.
You can adjust that now, though, right?
Yeah, we can.
Yeah, that's awesome.
What's that? No, I'm coming down below you on purpose. I need to be below you so I can turn around. If you want, here I can do it for you. All those are on. I'm going to click on your auto head. Yeah, that way. Yeah, until I get moved around. You can keep coming 25 or so. What happened to Herc's icon? It disappeared, didn't it? Herc's icon disappeared.
It's in the scroll.
There it is. We're on we're on DVL now, are we? Okay. Roger. Yeah.
Bottom in sight.
You can stop there and bring your head around to the north. Clockwise. Yeah, yes please. That is the dive salvo, but you can press it again. I just had the winch up there.
Mackenzie, can you do me a favor and push that salvo one more time and tell me if your control panel then says no response?
It worked good.
Okay, great. Okay, great, thank you.
here. We are late. Oh. 6666 66667. You don't need the res gauges. You can go off of the 99% there. I'm in auto XY, by the way, and don't reset the DVL or Herc will go to its new position. I rarely use it, but I use it now just to test it once every dive and
remember why you don't use it?
Remember why I don't use it. Exactly. Somewhere there.
Coming in. You need to get me one of those stream decks over here.
I do.
ED. All right. That's a great position right there.
Right.
I'm going to go ahead and black balance the camera. This will take it dark. This is intentional starting now. Oh, that's a good black to start with. Oh, that's even better. There we go. Oh, that looks like a good white too. White balancing starting now. Oh, no, that's better. Storing it.
So one of the ones that we are going to log on this one while we're below 3,000 is this tension. So the max we've seen, actually we took a 13 there. And I reset that when we were on the surface, so yeah.
I'll get on white balance, thank you, Dan.
Roger. And then after you log it, Mackenzie, on the hour, we'll hit this reset button here. You can actually log it as 12,999. Officially within the limit. 13 is bad, but we were still moving the winch then, so I'll let that one slide. Okay, looks like everything is working okay. All good there. Ennis got the longest checklist of us all. Niskins.
2,
right.
I think. Although, I don't think we have a port camera, do we?
We need to adjust it. I can see the back too. We're only going to take 2 on this dive, I think, right?
Yeah, and these aren't samples. I think they're background or to be used for cleaning of the things.
So currently we are taking some Niskin readings, but these are not samples. This is to clear the sampler. Verena, would you want to talk a little bit about some of our objectives? I know there's been some snail talk on the previous watch. For anybody that might be joining, what's the biological kind of highlights of this site that you kind of have in mind as we get started?
Just generally what we're trying to do is relocate the hydrothermal vents that we know are here. They're low temperature vents, and do a little exploration for possibly a site that's been missed in the past that might have high temperature venting. So we're starting south of where we know the original vents were, and we're going to do a little zigzag pattern moving north. And then just hop over to another small ridge that hasn't been explored and just might be the source of
I need to move that
the anomaly
so
that
I
was
can
in the water
see the
column
bottom of the other ones.
10 years ago.
Can you put a note in the red book?
Of course, have to remember it's 10 years, and so things may have
Just
changed.
adjust the HBU camera to the right a little, and we can see the bottom of the Niskins. I meant to do that. It's not in the log. It's not going to happen. Thank you. Okay. Science, we are all yours.
So first we're going to ask Jacob if he wants a rock here.
Yeah, I think this is a pretty good spot. If we could kind of see if we could get away a little bit from some of the kind of orangish sites, like right in here would be pretty good.
So the orange staining on the rock is iron oxide. There is or has been recently a very minor amount of seepage from vent fluid coming through here. And the fluid has no hydrogen sulfide in it. Instead, the iron in the fluid is forming an oxide rather than a FES.
They all look orange to me.
And if...
Yeah.
Actually, Dan, I'm also going to say that because of the staining, just without moving the ship, I was wondering if we could meander to the south.
Yeah.
The other thing that, yeah, Jacob, just keep your eyes out.
Oh, yeah.
I'm saying that because there's also the little white squat lobsters are here, and they are vent indicators. Now, we assumed that we would be landing with no vents down this far south of our target. So I just wanted to see whether anything just...
Looks like they're starting to get even more squat lobsters as we move in that general direction.
Oh, that's looking east there.
Yeah. Okay, and that's...
Whoa.
Looking south.
Yeah, and it seems to be up on this. Yeah.
Yeah, it seems to follow the white dots.
Kind of starts right there.
Yeah, what's just interesting to me is that this seems like it's kind of all, you know, on top of this older flow, and that maybe that when this lava cooled, it formed these short-lived vents that were iron rich and.
But it's still venting, right? It's
Yeah.
it's
They still have swell.
still putting
Yeah.
something that's.
Yeah.
And we have.
Well, that was 20 east. I know you said south, but I went where the white guys were. White things. Turn to the south here. I gotta do something. Can you come down 5 meters, Mackenzie, and pull and double tap your auto heading for me? Sorry, I'm dragging you around here. Trying to go.
Oh, it just seems to be kind of diffuse. Interesting corals too.
Yeah.
I'll go south and take the waypoint box.
Right. And we've died out here. Would this be a rock site?
Yeah. I think this would be a good one. These are definitely a bit older because you know, the corals and.
Yeah. Oh, certainly. Yeah.
Yeah. I think it would be nice too if it's possible to get kind of a sample of some of the younger stuff that we're seeing as we travel along it, if there is any.
Sure.
Yeah.
If you guys look on the lower camera here, there's something.
Oh
there.
yeah, some fish.
What type of fish is that?
Contact. Yeah, that one's loose.
Now it is.
You want one of those rocks, or you want to move on?
Sure. I mean, we're kind of after this crust right here. Anything that'll be sort of glassy or even like maybe kind of breaking off this piece would be good.
All right. Come down 5 meters for me. Oh, what we do here, poke, loose, but probably pretty friable. That one is not loose. That guy's a candidate. I didn't hear a no.
Oh, sorry. You just tapped this dude?
Yeah.
All right.
I think we can get that one.
Let's get a closer look at it real quick.
Go ahead, Ed.
I'll come in, get some focus here. I think it's pretty close. Yeah, there we go.
Yeah, definitely older.
Oh yeah, this stuff will be.
Yeah, I think that's good. I like the crust on it, so. Yeah.
Okay, Mackenzie, you know what to do. Starboard.
Then turn off port 2. There we go. Gonna zoom it will here on Zeus. Try to where it came from.
That light not coming on? Yeah, I think it was on and you turned it off. It's off right now. The camera one. Might have to refresh the page. If I can get it here. Go ahead. No. Let me toggle it here. Yeah, I didn't see it go on when, but I had it on earlier. Yeah, here, let me do something here. You have to go here and hit the Hotel West restart. This is our relay thing. And give it a second while I'm swinging around, then try and turn it on again. You might have to refresh your page too.
Like that's
yeah. So we've had a request. We've had a request online for something. I'll just get to you in just a moment.
From one of our scientists ashore.
Kind of big, huh?
Yeah, just gotta get the right angle of the dangle. I think it'll go.
Here.
There we go. Alpha.
Down here?
Yeah, I think that'd be good.
That might be good.
And if that doesn't work, we can always kind of venture around there.
Yeah.
You can leave that light on if you want. Yeah, you'll have to go to proportional control, and you know how to do that. Yeah, probably like 55%.
I think that's a question for, for Ed.
Yes.
Oh, Dan, before we head off from here.
Hold position.
Yeah, sorry.
All good.
Yes. It shows up on our nautiluslive.org website on satellite feed 3 and satellite feed 1. Yep.
Dan, the request is to suction some of the fine sediment, such as you see in here, and there was a pond. It's down in between the pillows or on top of the pillows with this sediment.
Roger.
Which
zoom in there real quick, Ed.
I
Yep, coming in.
suspect is pelagic
Say when.
sediment.
All the way.
Oh, these?
Those.
Counting to 8. Can everybody count to 8?
You want to switch cameras, Mackenzie, to the jars, and we'll rotate around to the red one.
5, 6. I got 7, Ed.
Really?
No, I'm sort of joking.
4, 5, 6, yeah.
2, 3, 4, 5. Actually.
Yeah, you're gonna probably have to adjust the hydraulics for that too.
Does look like an octocoral.
Okay,
yes,
you can go wide it.
Coming out. Quickly. There we
Sorry,
go.
we drifted a bit there.
Yep.
Not sure what's going on.
Yeah, we were a little bouncy when you were doing the sample too.
Yeah. I'm kind of far away from Atalanta. You can come down another 3 or 4 meters. So if you adjust your
Well, there's some right below you.
Yeah, I'm just gonna bring my head around so I just
There's
get
a
my
bunch of it.
tail yanked. If you adjust the jar speed up, Mackenzie. And we'll come around to the flush jar and give it a good go, and then I think she'll probably want one of the fine jars. I'll have to adjust the speed up, eh? It might be like slow the whole time. Yep, yeah, I think so. And turn on the suction now.
So we'll be putting...
Give it, slide that, yeah.
We'll be putting it into the suction jar 6 or 7 because the mesh is different.
Roger. Slide that PRV all the way up to 100%. Let me see something. What do you got going on here?
Check something here. Yeah, there we go.
Suction pump. Yeah. Yeah, so that one's 100, and then, yeah, that's full jam. That's all you get there. Yeah, just junk in the jar. And then go counterclockwise to 7.
It's moving slow.
Give it some more jam.
So this is still going to be finer than the mesh that we have on there. So perhaps not a huge blast. We retain some of it.
Try going the other way. I was probably stealing a lot of your jam there. I had it pinned pretty hard.
That looks like 7. Oh.
Come down another 3 meters. We're tail to tail, so it's pulling me around when we get the heave. Come down till it hurts. Yeah, you can see Herc there doing the wave. That thing doesn't, what do you got going on here? Proportional control. So, unless that camera is moved, it's a Geneva drive, so it should stop in the right place. Yeah.
I think the camera's panned
Yeah.
more left than normal.
Yeah, so that's another note for the red book. So it's if you know how a Geneva drive works, it's
Okay.
bulk.
So we've had a request from one of our scientists ashore to
Okay.
sample the sediments on
Zoom
the
in there, Ed, and
on these
Yeah,
pillow
stand by.
lavas.
give me
So
full.
that's
Yeah,
what we're positioned to
they
do
can
at the
see
moment.
that sample jar on satellite feed 3 on the website. I'm going to hold with the wrist. Oh, there's a bunch of little holes in there. See that?
Yeah.
Huh.
Might get more in your bargain for there.
Yeah.
Go ahead, Mackenzie. Suction on.
Okay. Suction on.
I'm seeing movement.
Herc is moving, so this could be. Yeah, that's okay. Go wide there.
Yep.
Find another spot.
There we go.
I gotta come back closer to Atalanta there.
You can go across the top of the pillow too if it's easier. So this lava formation that we're looking at, this is basalt, which is the most common rock on our planet. It's just that you don't get to see a lot of it because it's under the ocean. The formation here, these are pillow lavas. These form generally when it's a cooler lava that's coming out on the surface, and it comes out as these big chunky rounded pillows. You may have seen the videos of lava going into the ocean in Hawaii, where it quenched into these pillow forms. There are places we see lavas that have different morphologies, rather reflecting to some degree composition, but mostly the temperature of the lava, right?
Zoom in there, Ed. So that little pocket there.
There might be a bit of a reach with that those things sticking up on the right.
Okay, go in.
That on the left looks a little easier.
Definitely dusty here. Won't hurt. Find a home.
Some on that rock right in front of you too.
What's that?
There's some on that rock right in front of you that you're stretching the hose over right now.
I'm gonna zoom in there.
Yep, zoom in. Holding.
Turn the seconds.
Sections on.
Right.
Not really seeing movement in the jar. Right now there's some movement.
I have a feeling it's going right through the mesh.
Yeah, it's very fine.
Turn the PRV down to like 50.
Okay.
It'll clog up the mesh here eventually. That's 50, is it?
55-ish.
Roger. You'll know when it clogs up the mesh when it stops sucking. Your vacuum is full. Time to change the filter.
You know, I could be imagining things, but I think as you're sucking up that sediment, you're getting some glacier bits of rock too.
Yeah.
Yeah, which I don't mind.
Good. Okay, maybe we could see what settles it.
Sure. Suction off.
Okay.
Okay.
It's not a lot of sediment, is it?
Well, try this. Trying to knock it off of the immediate. You can see it falling down off the.
Yeah, off the
But I don't know
filter.
how. Yeah, I don't know how fine it is.
The filter is a 500 micron.
All right.
So, and this is very fine stuff.
We could try some more on a lower setting if you
Yeah,
want.
try that and then turn the suction off before you pull the hose away with clear water.
Can we rotate while we have suction on, Dan?
No.
No, we gotta
Okay, try
get the sample in.
25%, Mackenzie.
Okay. 25%. Ready for suction?
Yep.
Okay. Suction is on.
Seem to be picking up anything there. There we go. Sort of get the angle of the dingo right. So sorry, you wanted to turn it off while I'm still
Just
in
so
the
that
mud?
you're not pumping fresh water through, clean water through and just flushing what's in there in the line.
Oh, yeah, yeah,
You know
yeah.
what I mean?
Yeah. Usually I have the opposite problem. I plug this thing like picking up dog hair at my house. We have 2 German Shepherds.
Okay, that's got a bit of a storm in there. Yeah.
Okay, turn it off, turn it off, turn it off.
Turn it off.
Let's say that jar is full of sediment.
Okay, let's say it is. We'll see what comes up. Okay. So we kind of ran out of squat lobsters. We can turn around and go back and start our northerly trek.
Roger. You want to go the other way to the suction jar because at some point we'll have to blow all that junk out before we get to the next sample.
Verena, did you say you wanted to head to the next waypoint?
Yes,
Turn that
let's
back
do
up
that.
to
Okay.
Thanks, Anna.
something like that.
Do you have a flush jar?
We do, that one right
oh,
there.
that red one, yes.
Okay, you can bring your head back to the north. It's going to take half an hour for Atalanta to move, so you can get the ship moving if you want. Or I could do a long move.
Yeah, it's about 50 meters away, so
Yeah,
we'll do 50, yeah?
sure. The danger here is if there's any cliffs in our future, we'll have that layback.
Yeah.
Let's time it just for fun. From
Okay.
when you start
yeah,
to
yeah,
move
to when it starts to move.
when
Yeah.
it stops. That's good to know because you'll get a feel for
Yeah.
what's up.
Bridge now. Hi, can we have a 50 meters at bearing 325? 325, yeah. Thank you.
Log in, put bird dog in there. Oh wait, here it is. Kicking the NDI in the guts.
Copy. Yep, they're gone. No.
Is that in the water column there kind of floating down? Is that just...
That's from the suction sampler.
Okay, that's our leftover.
Yep.
But but then sort of the smaller bits, is that marine snow?
Oh, the white bits. Well, I've been wondering that myself because it does seem fairly flocculated, and I'm just wondering if it's coming off
terrain in Atlanta.
some vent.
Maybe, yeah, escaping the vents.
Yeah, I see it. Suction's not on, is it? I've got no jam here.
Oh, your exhaust.
Maybe that's how it got spread out all over the place. Somebody came down and reversed their suction. Unlikely.
Especially because most suction samplers use a Venturi effect, which I don't believe can be reversed.
That reminds me of a movie that I'm not going to talk about because we're doing science here, not talking about movies anymore. How about the bamboo corals we're seeing? Pretty spread out. Do you think this used to have a higher density for that bamboo colony, or is this kind of what you saw in the previous dives at this site?
We're not seeing a lot of skeletons, unlike the dive we did Boat Seamount, where there was tons of skeletons all over the place.
Yeah.
And
that's
a beautiful anemone.
Yeah, there are a couple of anemones there.
Two of them.
We are headed to a place called Anemone Heaven.
Okay.
Uh-huh. So I think this is sort of normal deep sea, sparse.
Yeah.
Making a living as best they can. Maybe a little extra input from a vent every now and then.
Are they back yet, Ed?
Yeah. Yeah, lower came right back. Like right back.
I feel bad for those anemone. They're so close to anemone heaven.
Yeah.
They didn't quite make it.
Purgatory.
Knocking on anemone heaven.
Yeah. They're on their way.
And it seems like we've gotten away from all those squat lobsters.
Yes, no, you're right.
Sorry, what do you see?
A ton of looks like associates on this. Well, it's hard to tell. Or maybe part of it's been eaten. Oh.
Yeah, it's got zoanthids on it.
Yeah. Huh. It's the first one of those I've seen.
See if I can get my camera lined up with the stills camera there. Push in a bit, Ed.
Yep, coming. Holding.
I need to get out of, I'm in full throttle mode here. I need to chill out.
It's amazing that it can support so many. Doesn't pull it down or something.
And there it goes.
Off we go.
Sea star or carnoid back there? Just had a frame top.
I don't know. I was looking at the sky here. They're always fun to fly around. Bonk.
Double bonk.
Nautilus is starting to move here. And what was our bearing?
320, 325?
325.
Right.
Yeah. Took 11 minutes for Atalanta to start moving.
Yeah, the scary part is it takes 11 minutes for it to stop and there's like this red wall of death coming in the sonar. And there's nothing you can do but panic and come out, and like the wall's full of beautiful biology and geology, and you'll never get to see it again. Because it would take like a half an hour to move back.
This is quite heavily sedimented here. These are pretty old site.
Another one to get to know is what the layback is. So, like the boat was here, we had hardly any layback. And now that Atalanta's moving, we have the 40, 50 meter layback, as we call it. So the separation between from where it normally hangs straight to where it started moving.
You can see how the lavas have rolled down the hill. Just the tube one after another.
And some squats again.
Was that a polychaete in the bottom?
No, that was a squat lobster. He's
Oh.
swimming.
Oh.
If you want to see anything closer in the back row, let me know. Otherwise, I'm just going to
No, just keep transecting.
Yeah, keep transecting.
Do we know what species that squat lobster is?
Well, it's one of two. You remember you looked at the two back arc ones? So
yeah, that's Munidopsis laoensis?
Marianus?
Marianus?
Or myogenensis is what, yeah, that's what you're, yeah.
That flesh jar is still full of.
Yeah, it's still settling. So there, I guess we got some. Oh, that's the flesh jar.
I turned it on for a minute and it sucked all the stuff that we had in the hose into there, and it's.
Yeah.
Then I turned it back off because it steals a lot of our power. You can change that camera back to the rail cam if you want. I might have to put my the rail cam back up. I really miss it. we would have had that, we might not have had the avalanche. Yeah, that's my story, and I'm sticking to it.
The avalanche was a blessing in disguise because it gave us the chance to fix the rear bumper.
Yeah, and I get to order more speed rail, which is fun to make stuff out of. So we should set our WCA cameras with the bottom one zoomed in, the middle one zoomed in probably 40–50%, just so you can barely see a hint of the bumper. And the top one is in. You want to turn on the suction for a minute? They're good.
Okay.
You still see stuff coming out of the hose there. Look at the exhaust pipe there.
Yep.
Rolling coal.
That's probably good there. It's got clear water now. Thank you. With these, I try and keep about 20 meters, 10, 20 meters out in front of Atalanta when we're cruising like this. And we're moving it 10 meters a minute. That gives, you know, about 2 minutes to get a sample or look at something before Atalanta swings in on top of us.
Yeah, we're down quite a long way here, 3,700 meters. And you can see the seafloor is not exactly crammed with life here. It's a pretty sparse living. Most of these coral, well, the corals basically just have to wait till something floats down from the surface or some small deep sea crustacean gets tangled in their tentacles. So it's a pretty hard way to make a living.
And isn't that, Verena, I heard some crazy number for how long it takes for particles to float down to this depth.
You know, there's been a lot of calculations based on drag and the size and shape of various particles. So it can take years and years. But then, to be honest, most of the way that particles get to the bottom of the ocean is that they glom together, or they've been through the gut of some copepod or shrimp or something in the water column. They fall a lot faster than those single tiny little particles with a huge amount of drag.
So I've ticked the waypoint 2 box.
Yep.
You're probably watching back there, I guess.
Yes, see that. So we're doing a zag now.
Next waypoint's like 040.
Sure.
This way, yeah.
Yeah.
90 meters.
Sure.
Just do it in one.
Yeah, why not?
Okay.
.040 for you too, Mackenzie. you can, I'll come around in front of you so you can kind of track me around.
The Mariana back arc is a very slow spreading center, although in the
Or
south
an
it is.
eel.
It's only about 2 centimeters a year.
I'm going to say eel at this distance.
It's got a,
Well, it
looks
could be
like a
fish.
rat tail.
Yeah.
And
Hard to tell.
I've never seen one of these things
oh, maybe you're right.
eat anything.
Maybe it is a...
When they do, it's a
eel.
ferocious burst of energy. They go from this very conservative movement to, I need to eat that immediately.
They have to save energy.
Yeah,
These
it's
things
cold,
are,
not a lot of food. Huh, I don't think I've, oh, maybe I have.
You push in just a little bit there.
Just a little push. And holding there.
Get the Zeus pop.
Look at the...
That's pretty healthy looking. I don't see any parasites or
Look
anything.
at the back,
Yeah, that's pretty cool.
Yes.
That's big.
Yeah. How old do you think it is, Verena, if you had to guess?
I'm sorry. They do live a long time. They have very, very slow life. Very, very low metabolism, because they just have to conserve it, as Ed says.
Can I do a push here?
I'll get closer here.
Yeah.
It's cooperating nicely. I'll let him swim out of the frame.
Yeah, let him go. Let her go. And coming out.
Nice thing set.
Thank you.
How much do you think a fish like that has to eat to maintain that size? It's got to be collecting a lot of food.
No, it's a matter of metabolism, how fast your metabolism is. So it's not so much the size, it's if you've got a very, very low metabolism, and he's living at 2 degrees centigrade.
Right.
And so everything is just very slow. Just so you don't need to find that much food. You do obviously have to find enough to keep going. There's been a fair bit of work on the metabolic rates of grenadiers, which are similar rat tails, which are similarly deep sea. You can get species in the abyss, this kind of depth, and then species that live up on the continental slope at, say, 1,000 meters, and they've compared their metabolic rates. Very different, because the guys on the continental slope can get a lot more food.
Right.
They move faster, they eat more. In the bottom of the ocean, really, really slow, long lives. They don't reproduce for a long time. I don't remember the numbers now. I just
Kind of.
So the oh yeah, go ahead.
To change the subject, I think I'm seeing some hints of
little bit of
venting,
a little
yeah.
hint of
Just a
venting.
heads up.
Yep, and
Yeah.
the anemones are getting. These are the anemones that are the vent ones. The one we saw before actually probably wasn't.
And this is starting to look like younger lavas
Flows?
as well.
Yeah, you haven't got as much sediment on it.
Yeah.
No, that's a good point.
Interesting. Look how altered they are. Yeah.
So this anemone is called Marianactus, after Mariana. Okay, that's
Come
good.
down a couple meters for
Good
me.
sign.
Find something to look at while I wait for you. Bit of a breeze here was pushing me faster than
Yeah, this is looking very young.
Yeah, it's interesting, that transition that we went over.
Dan, do you think it'd be possible to sample maybe somewhere off to the left, or?
yeah.
All right.
Right off the zigzag pillow there.
Yeah, yeah, that'll work.
Come down a few more meters for me, Mackenzie.
So what happened here was that we were traveling along looking at pillow basalts that were covered with sediment, which means they're pretty older lavas, but all of a sudden we went over a transition where there were newer lavas, and we're not seeing that pelagic sediment on top of them.
You can really start to see those anemones too there, especially in the fish eye. You can see it's quite plentiful.
There's an anemone there with a slightly different pigment.
Yeah.
Suggests that he might have had a different dinner. I don't know.
And I have to overexpose those anemones so we can see the rock. Fortunately, we'll image some at some point.
Yep.
Oh, what's going on here?
Huh.
It looks pretty solid.
Mm-hmm.
Negatory goes right here.
Yeah.
Yeah, we'll just keep going around, and if we see anything that looks good, you know, we can drop in and grab some.
Right. I'll have to. Yeah, we're plenty of tether in the bank as it were at the moment.
Yeah, maybe that one.
If I can get down there to reach.
I'm gonna
Chop for that rock. Well, that was not very elegant.
There's a grab and go right here.
All right, let's see how it looks.
In a bit more of a hurry because the vessel's on the move.
Oh yeah,
Glass, baby.
that's fantastic.
This is the
Go
most
zoom in
excited
there,
I've
Ed.
heard you for a rock.
Yep, coming in. Stand by right there. And
Yes.
focus.
What are you seeing on that one, Jacob?
All right, it's all glass. It's really sheeny. It's yeah. Let's get it in the. Oh my. This is very happy.
Okay.
Coming out.
I know what to do, Mackenzie.
Here.
Who says geologists
thought
aren't romantic?
Comment back in here. We've got some little vent animals too, but we won't worry about them right now.
Oh yeah, I see them.
You can zoom in on them.
I think
They're way off to the left.
Yeah, it's okay. I think they might be barnacles.
I see shimmering water there.
Come in here and do a little something, something.
Oh, I see shimmering water.
Yep.
Yes, I see it too.
Pan up and to the left for Ed, Mackenzie.
Kind of. Where's T? There it is. Like over in here.
Oh, there's the, yeah, either one. Yeah.
Right there.
That's about as far
I
as
can
the camera goes
see
left.
the shimmer in there.
Yeah.
Yep.
B.
Yeah.
Oh boy, look at that rock.
Oh yeah, right in here.
Yeah, yeah.
Okay, you can close the box too.
Anyway, just a taste of what I hope is to come.
I'm going to pop out here since we're moving.
Yeah,
it hurts.
Nope.
And go back to dive. That box is moving slow.
Next to the vent there, you think that's
everything moves slow down here.
Or that's something else?
It could be.
Yeah.
It's kind of exactly the right habitat for them.
Could that be kind of what we're seeing in the water too that's falling down? Some of that venting, I mean, just this, it almost has like a reddish orange hue.
I'm unsure.
Yeah.
I'm not sure what these particulates are.
Pretty good shot in Atalanta of all these organisms up here.
Mackenzie has absolutely no fear. Right on the deck.
Yeah. Riding on the back of Herc.
Dr. Ballard would be proud of you, Mackenzie.
Yeah. Wow, this thing's tall.
That's a really
Wait, where's the
tall
end of it?
coral.
Yes.
Tickling the upper camera.
Yeah, it's in the
It just
thruster.
keeps going.
The beanstalk. Wow. Really?
I can see
What's
it.
your altitude?
I can see it in the upper camera, 4 meters. And it's still.
Still going. Yeah. Wow. How about now?
5 meters.
5 meters?
And so you can just see a hint of the porch there in the Zeus. So
Yeah.
5 meters tall. He's a big one.
Oh, Lord. Don't do that.
I'm seeing right to left current in the focus monitor. Oh, maybe you can see it as well.
Seems to be switching around on me. Cause
Yeah, it was the other way.
Yeah, it was, totally. Moments ago.
Fair diversity of Cnidaria here. Yeah, between the anemones, the corals, they're all in the same phylum.
It's interesting you had that high density of anemones where we got that rock sample, kind of where the shimmer was. Maybe something going on there.
Yes, these anemone, the white anemones are only found on the periphery of the vents here.
Yeah.
Put this one in
they seem to be particularly
in the still camera.
They're more abundant on this.
Yep.
These sites
looks like
in the northern
anemone
than we've seen them down further. Yes, this is an anemone.
Yeah.
On the end. That's pretty funky.
Totally.
Yeah. That is the proper term for that.
That would be a nice. Oh, you haven't got them aligned, have you?
Oh, of course.
Maybe you could get them in the still cam when you're done there.
Yeah, he was, but yeah, I could. I'll have to back up a bit. Come on, Herc, float.
And come out while you get in that camera.
Yep, good.
Got it?
Thank you.
Yep.
What do you make of the structure of that anemone? How is it able to, is it on the end of that coral or is that stem part of it?
No, it's on the end of the coral. Anemones have this thing called the pedal disc. So it's just a flat, fleshy bottom which just integrates,
You have to come up a
all
bit,
the
Mackenzie.
flesh sort of
Single
integrates with the
digits there now.
substratum, but in this case it's just wrapped it right around the end of the coral and around itself. So just tightened on like a giant suction cup all the way around. So here you can see the pedal disc at the bottom. Ooh, I gotta do this. Shh, that.
So has that anemone been at the end of that bamboo coral when it was, 10 centimeters long? And
My
then
guess not. My guess is that the coral is grown up and then the little larva came along
settled.
and didn't get eaten. Maybe something else had nipped off the end of it and killed the end of the stalk. I mean, I can't build a scenario, Sean. I don't know.
It's definitely out in the water column.
So we've got more iron staining here as well. And I'm finding it, interested that the corals are relatively
Bring
abundant.
your head to the right just a bit for me. Let's see that toilet.
I suspect a lot of this was active venting when we had a younger lava, and now it's just losing a lot of the hydrogen sulfide, so it's turning to iron oxides.
You come. Oh, no, you're good. You're good. I need to come uphill from you. I'm going to come up the hill, so on the other side of it. I should be on the north side of you there, because the slope's on our left there.
We're gradually climbing a small ridge.
Yeah, I'm taking a zag here to the northwest just so I can get north of Atlantic. Mackenzie's got that kind of steep hill.
That's a zig.
Oh, sorry, a zig.
Northwest is a zig.
Roger. How did I always get east and west backwards? So zigzag east west. Zig left. I should remember that. I kind of fly right by you there, so because you're just 11, you're single digits off the deck now, right? So yeah, you should be able to turn left and come up as I should be able to go north now.
See how these pillows have cracked when they've cooled very rapidly.
That one causes them to crack?
Yeah, they start to contract.
Similar to that Hershey's hard shell stuff that goes on ice cream. Is it Sunday yet?
I was just thinking the same thing.
Oh, it's actual Tuesday. We're nowhere near Sunday.
So that's better. Your delta's par with your altitude now, right? But it should be more altitude than delta if Herc's on the right side of you, or if Herc's where it should be. So we'll keep going until we get. Then when we stretch out, you can come right down to par
there's
delta
flow
without
right
hitting
there.
the seabed.
A lot of shimmer below the lasers.
Yeah, starting to get into younger flows again.
Yeah.
What's that?
There's just a lot of flow down that, that dark cavern to the right, and just below the lasers, like right in there, you can kind of see it.
Yeah, the white stuff.
See it in there?
Yep.
Yeah, shimmering.
To the right, even more. Just to the right of that. Yeah, right below there. That's where the most of it is below that.
Well, there's no telltale, like no bacteria, no
Nope.
animals, no nothing. Maybe it's not, you'll have to come up a bit more, Mackenzie, I'm right into you. Tether's bouncing there.
Oh, there's our first
do it.
Astinagria.
Mm-hmm.
So this is the counterpart to the Gandalfus crab.
Ah.
Looks in the same Bythograeidae. Just as there's a different squat lobster on the arc, and from the one on the back arc that we're looking at, so there's a different crab.
Have you sampled that species before?
Yes, it was collected actually, and it first described from the 87 collections. Astinagria after Austin Williams.
Ah.
So Astinagria williamsi.
Ah.
So yes, that means something to you, yes.
Yeah.
He was at the Smithsonian, right?
Mm-hmm.
Yeah. It was Bob Hessler who named it.
Oh, okay.
What's our next one is a zig, is it? That'll be nice up the hill.
Waypoint 3, okay, so you're headed to Anemone Gardens, are you?
So, yeah, we're 30 meters from waypoint 3, but the boat stopped, so we could go north from here.
Yeah. Well, maybe we could just go northeast to get over, just because I know we haven't been there. I know that's a northwest.
Yep, we could do a northeast move.
And then go west over to Anemone Gardens. Let's just skirt around the edge of the ridge crust there.
How far do you want to go northeast?
What's your scale there?
My little...
this is, can you see the high pack?
Yeah.
Yeah, it says 45 meters.
So from where you are now, if you went 40, it's 45 meters in diameter or radius?
Radius.
Okay. Yeah, if you went 45 meters
45
in northwest.
Yeah.
That'd be good from where we are.
Northwest or northeast?
Northeast. Huh? You know, you and I are the same. East-west, yeah.
We'll call it a zig.
Oh, that's a zag.
No.
Okay,
yep,
then
he's right. It's a zag.
I didn't know those had cardinal directions associated with them.
They do.
Okay, so let's get it straight then. So zig is...
Zig is left, and zag is right.
And zag is right. All right. Zig, zag.
It's Anna's job to interpret what people say in the opposite direction.
It's like a great game of telephone.
Yeah, so if you zag 4 times, you go just running in circles.
Right. Zag. Not cubed, but one more time.
You can bring your head to 0452 for me, Mackenzie. Be great.
Oh, look at that. That is a hydrazone.
Push in a bit there.
Yeah. Flow dry by here real quick.
It was spotted here first as an unknown by the OE dive here in 2016. Ah.
Come down a little for me.
And I wrote it down in my notes up in my cabin. Oh, and there's a baby one.
Two, three, yeah.
Isn't that gorgeous? It's in the benthic guide. Hydrozoa. Let me see if I can find it.
Never seen one of those before.
Yes. Oh, well, if we can give you something new.
Yeah.
Hydrozoans other. I think I'm right. Oh, that's just stunning. There it is. Candelabra-D.
Huh. Appropriate name.
Is
Could do a quick
just
push.
beautiful.
Just get
Hydrazone.
60 seconds there or so, and then
Oh, and
Gonna
then
do a
just the fact that it's got a
quick
1, 2,
zoom for
3.
detail, just to get some of this.
So they're very distantly related to corals. These are probably little stinging tentacles on it. Can you
And
go
then,
over to the little ones on the right as well?
Yep,
yeah.
stand by. Gonna try and get these real quick for you.
Yeah. I wonder if they bud off.
Look at the base of the
Yeah, the
larger one.
cluster.
Because that's not been seen before.
That's good.
Oh, I so hoped we'd see one of those.
Losing my focus. There we
It's
go.
no species name, it's just unknown. So Candelabra-D,
And one zoom here.
Candelabra-D. There's a slightly different one seen from the Johnston Atoll, and then the OE expedition saw this one here, a little way south of here. Oh, that's nice.
What is it at the stem that's happening there?
At the bottom, the little...
Yeah.
I don't know, but it's probably a reproductive structure of some sort.
Is that one, the small one on the far left, attached to the body, or is it already attached to the
There's a big
bottom?
one. Say again?
I don't know.
Gotta go?
Yeah.
Nope, we're good.
No, it looks like it's on the rock.
Down.
It's on the rock.
A little ways up its body, it's almost like it has a piece branching off.
What, this?
Yeah.
Yeah. I wonder if it buds.
Then attaches right next.
Right.
Not far from the stalk.
Right.
Oh, that's lovely.
Gonna do
See, it looks a little different from
slow
this
pull.
one.
Right. Wow.
See if I can do a super slow pan while you're
Yeah.
Anybody have an estimate for how many of those
Nice.
spikes are total?
I'm gonna hold here because I don't know where the housing is. Got tether.
What's that?
Tether's bouncing on. Oh, actually, no, it's
Yeah,
fine.
that's
It's fine. Just caught it out of the corner of my eye. It's gonna come out just a little bit more.
While we're here, just gonna float right up and get a shot of the critter above us.
Yep.
Okay, you can come up a couple of meters now, nice and easy.
Wow. I think I'm still partial.
Yeah, you are. That's all right. Did you get that, forget what you called it, the wavy thing in the, with your still camera?
Oh, yeah, let me get another shot.
Yeah, I,
Oh,
sorry. I'll back off and slowly
Were you able to
float
get
up
the
here.
babies in the still camera, or are they too small?
no, you can see the babies in the still camera a little bit.
Nice.
There we go. And one more. Perfect.
I'll just float up and you can keep clicking if you want.
Got it.
Maybe I'll float up.
So you
Okay.
mentioned.
Is that another one up on the top to the left?
Looks like
Oh
it.
yeah.
Yeah.
There is another one.
Oh.
You said they're distantly related to coral?
There's one to the right.
They're all over.
That's okay.
Quite distant. The cylindrids are in 3 major groups.
Wow.
The group that has the corals in it are the anthozoans, and then these are in a whole different group called the hydrozoans, which include things like the Portuguese man-o-war, stinging coral, and it's not, stinging coral is not coral, it belongs to, so they've evolved in different lines, but they're related, let's put it that way, in the same phylum.
And do you mostly find them in deep sea environments?
No, they're up on coral reefs too.
Yeah.
They're in freshwater lakes, little tiny hydroids, or coral hydrozoans. Actually, the anthozoans don't go into freshwater, but the hydrozoans do. I think that's correct.
So I've been trying to restrain myself a little bit and give
Good.
the bio a chance, but
I was going to say, can you chill out over there a little bit?
I was hoping we could pick up another rock sample if we get a chance soon, kind of from this older area.
Yeah. The rock
Actually,
right there next to the,
yeah, you thinking that one?
yeah.
Yep. That's good.
It'll be able to zoom in on the IM Sauron while we're putting it
Yeah.
on. I can get the minute out
I
here.
gotta get me a Steam Deck over here.
Stream.
Stream. Sorry. Steam Deck's another solution.
Yeah. I can hook you up.
They make a smaller one? They make all different sizes.
We happen to have a smaller one on board.
Oh, wow. What's that going to cost me? More iced tea, I'm guessing.
Something like that.
You want a little bit of zoom? Or you
All
good?
right. Let's zoom a little.
All right, just quick zoom here. And I'll hocus focus. There we go. 285 subsamples.
All right. I think we're good.
Great, coming out. And then we'll go to the sample over here.
Kenzie, you want to move the HD camera around for Ed to zoom in on that critter there?
The anemone lower left.
What's that?
Go ahead, continue opening your box. And then once it's open, if you could pan the HD camera left and down.
You might have to turn the PRV down. I had it cranked up.
That's good.
Somebody in the chat said, hashtag Team Jacob, more rocks, all caps. Which is making me think of the Twilight DVD that's sitting upstairs in the lounge.
Oh dear,
Yeah.
no.
I know, we're not going to go there.
Too far, Kyle.
Yeah, sorry.
So what are we up to now? C?
Yes, C.
I don't know if that one will go in C. One way to find out. Oh, bombs away. Ooh, that's too hot. I can see.
Yeah, that's 63.
Rock clear.
That's a nice shot. Beautiful flower-like pattern.
Hercules is about to get the Yankee from Atlanta.
Looks a little bit like when you slice a red onion in half.
Well, it's an actinostolid. That's about as far as I've got. Oh, I like this one. Actinostolidae, brown.
Quotation marks, brown.
Yes.
Okay, off we go.
There was the ID.
Sorry, I can hang out.
No, you're good. You're good.
Oh gosh, they all look so.
There you go.
It's got fairly long
On our way.
tentacles like that. It's a lot like that. Just.
Seem to be stuck in the rocks here.
Yep, there's another one. Northern Mariana Island.
Ooh, is that what I think it is? Push in a bit there, Ed.
Yep.
Oh, I always get buck
Oh.
fever when we find these.
Yeah. It's going to retract as soon as you get close.
No way.
These are so hard to image.
Oh,
Oh, easy,
forgotten
easy.
what those are. Are they siphonophores? Or is that...
Atlanta's past us.
Bentectinophore? Right? Oh, fishing lines out. Very fragile. That's the wrong one.
Are those...
Oh, it's got something there.
It goes retracting.
Yeah, it looks like it. They always retract when you get close to them.
Looks like the species is Lyroctis? Lyroctis is the genus. Don't know the species name exactly.
Oh, there he retracted. You predicted it.
Must be the pressure wave or something.
Yeah.
Is it using it like an anchor? It attaches.
It's a way to collect food.
Okay.
Long sticky stringers, and they collect.
So almost like a spider web.
Yes. Looks really cool.
Yeah. Can't quite make out its...
You got a little more push if you want, Dan.
Sure. Hands off the sticks here. Float, Herc.
All right.
Oh, I'm caught in a rock.
Okay, we could head towards Anemone Gardens.
Right.
All good, thanks.
Pretty much there. I totally get buck fever when we get around those things. I'm gonna start shaking. Usually a choke. That, and what's the other one that puts out all of its webs and anchors itself? Maybe that's the benthic ctenophore, and that one's something else.
I don't know if I've only ever seen them scoot. No, I think I've seen them with ROV. The Medusa worms, the ones that are under the...
What do we, our nickname for them, so like a sunstar or sea star or sunstar.
Brianna, did you get the name of that last creature?
Yeah, the genera, or the genus is Lyroctes, Lyrocteis, but I don't know the specific species.
What's that common name is?
Comb jelly.
Comb jelly.
Yeah,
Yeah,
that's
that's
right.
it. Thank you.
Nice.
Yeah.
What? So it is a ctenophore.
Yes. Yes. Yeah. I was wrong. It wasn't. But what Dan was talking about is a siphonophore. The little balls with the long spikes that attach to the seafloor.
Yeah. They put out a spike all the way around, and they're usually down in a hole that's like impossible to image.
Oh, there's another one. Yeah. Amazing.
They get quite long. Do they retract or extend out, you think?
No, I think they just wave around in the breeze. I don't know. This is the first time I've ever seen them.
Yeah, yeah.
It's just an indicative of current. That's what it does.
Right.
They look like that thing with arms you see outside of a car dealership.
Yeah, did anybody have that as a dance move they were going to do if we found one? There you go. That was Ed's. Dan's doing the spider dance. Ed's doing the rental car blow-up machine.
Hopefully we see the spider dance soon.
Any minute.
Yeah, Dan bolting for the door.
Yeah.
The one time he refuses to grab something.
Right.
We are pretty much at waypoint 4.
No, isn't that? Oh, yeah, right. You're getting there. Yeah. Well, we'll see. I thought it was a little further up on the top of the ridge, but we'll see when we get up there.
It's a steep hill.
Oh, that's Anemone Gardens. It's different from Anemone Heaven.
Although we are at the waypoint, it seems like we're actually seeing less anemones than before.
Well, that's a delta.
Yeah.
It's an observation.
Negative delta.
Actually, Anemone Gardens was waypoint 5.
Oh, so we haven't got there yet.
Yeah.
Find something to look at while waiting for the boat. What's that? You're muted. I can't hear you through these. Yep. Yeah. Yes, please. Yeah. You're good, Mackenzie. You got like 30 meters underneath you. I'm stretched out. Sorry. I'll come back down under you a little bit. Turn and get a sense of how steep it is here.
They're all over the place.
That's the same one?
Yeah.
Mm-hmm.
Huh. There's 3 of them there.
4.
4. Yep.
5.
I don't see the 5th one.
6.
Oh, wait. Oh, yeah.
I'm not sure about 6.
Yep. 7, 8. It's babies.
Yep. Oh my gosh, they're everywhere. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10,
Yes.
11.
Okay, does that count as a chimney over there? On the right, it's almost kind of pillar chimney-like.
Nope.
Probably not. This is also just
Nope.
the same. Yeah.
In your dreams,
Yeah,
Kyle.
yeah, yeah. Man.
You'll know those chimneys when you see them.
Yeah.
If that top one gets long enough, it might get tangled in that anemone. Those are pretty cool.
It's like 10 minutes for each zig and each zag. Wonder if they plan that into the dive plan. Ten minutes is not bad. If we have
There's
current,
another
it
one.
can be like twice that.
Right there. They're a dime a dozen.
Yeah, they are.
I'm assuming these have been collected as well.
Well,
I mean, would we want a sample of?
actually, I really, yep. I
I
really
mean-
would like to get snail pit. I think it would be good if we could try and do the vent thing, and then we see them again. We can do it.
Because they do seem to be quite plentiful.
Yeah, they do.
Is there a vent in our future? On this watch?
Was there any, in our mapping imagery, did we see any